Hello students, welcome to EPG Pathshala. I am Dr. Vikas Jain from BS Anangpuria Institute of Pharmacy, Faridabad. Today I am going to talk about module Monophasic liquids, its introduction and solubility. This particular module has been taken from paper Product Development 1. Learning objectives of this particular topic are monophasic liquids as pharmaceuticals, advantages and disadvantages, classifications and subtypes, solubility phenomenon, and various factors that do affect the solubility. What are monophasic liquids? That is the basic question. In pharmaceutical terms, monophasic liquids contain one or more soluble chemical substances dissolved in a suitable solvent. It is primarily employed to enhance the bioavailability of a therapeutic agent. It eliminates the need for a patient to swallow a tablet or capsule, hence remains acceptable for children and for elderly. What are the various desirability associated with various liquids? So, for this particular question, a simple answer is that it must satisfy the requirements of pharmaceutical elegance with regard to taste, appearance, and viscosity. So, now what basic features should you consider while formulating liquid preparation? Generally, these are intended to be administered either undiluted or after dilution. They may contain certain auxiliary substances like dispersing, emulsifying, suspending, weighting, solubilizing, thickening, stabilizing, and antimicrobial preservatives. It may also contain suitable sweetening, flavoring agents, and permitted colors. It may also be supplied as powder for reconstitution, that is prepared just before administration or use. So, while we are utilizing or administering powder for reconstitution, what are special considerations that are to be taken into account? First thing is, diluted oral liquid should be freshly prepared, irrespective of nature of the diluent. It should be used within the period stated on the label, because diluted form may be less stable physically and chemically than the corresponding undiluted preparation. It should contain suitable preservative, effective for dosing period. That means during use by patient, it should not permit any kind of degradation. Although we are going to discuss various categories of monophasic liquids under this module, major emphasis will be given on development and formulations of oral liquids only. Let's discuss the advantages of monophasic liquids. Liquid formulations offer many advantages over other dosage forms, like ease in dosing, to ease in administration, and myriad possibilities of innovative drug delivery system. It is suitable for pediatric and geriatric patients. It enhances the oral bioavailability of a drug. Absorption in solution form is more rapid, and it also provides ease in dosing and administration. Let's also discuss the disadvantages of monophasic liquids. The formulation of solutions presents many technical problems to industrial pharmacists. Some drugs are inherently unstable, and this property is magnified when the drug is in solution. Special techniques are required to solubilize poorly soluble drugs, and the final formulation must satisfy the requirement of pharmaceutical elegance with regards to taste, appearance, and viscosity. Therefore, a unique set of consideration is required during the design and formulation of monophasic liquids. Monophasic liquids can be broadly classified in four categories, that is, oral liquids, cutaneous preparations, oromucosal preparations, and miscellaneous. The first and foremost important category is oral liquids. It includes elixirs, linctuses, drops, and solutions. So, what do you think? What are elixirs? These are clear, flavored oral liquids containing one or more active ingredients dissolved in a vehicle that usually contains a high proportion of sucrose in a suitable polyhydryl alcohol or alcohols and may also contain ethanol up to 95%. Next category is linctuses. Linctuses are viscous oral liquids containing one or more active ingredients dissolved in a vehicle that usually contains a high proportion of sucrose. Next is drops. Oral drops are intended to be administered in small volume with the aid of a dropper. And next category is solutions. Liquids containing active ingredients dissolved in a suitable solvent. It can be oral rehydration solution or oral colonic leachate solution. Other categories under oral liquids are syrups, tinctures, fluid extracts, and aromatic water. The most important of these oral liquids is syrup. These are viscous oral liquids that may contain one or more active ingredient in solution. The vehicle usually contains large amounts of sucrose or other sugars to which certain polyhydric alcohols may be added to inhibit crystallization or to modify solubilization, taste, and other vehicle properties. Sugarless syrup may contain sweetening agents and thickening agents. Next category is tinctures. Tinctures are alcoholic or hydroalcoholic solution that may contain alcoholic content from 15 to 80%. It is prepared from vegetable materials or from chemical substances. Another category is fluid extracts. These are concentrated alcoholic solutions used for animal or vegetable drugs, generally prepared by maceration or percolation. And the last categories that has been used for labeling purposes is aromatic waters. These are saturated solutions of volatile oils or other aromatic substances in water. Cutaneous preparations under monophasic liquids. In this category, four major type of dosage forms are included that do include collodions, liniments, lotions, and topical solutions and tinctures. Collodions are liquids usually containing pyroxylin in a mixture of ethanol and ether. After application and drying, a flexible film is generated at the site of application. Liniments, as we all know, these are alcoholic or oil-based solutions containing topically active therapeutic agent intended to be applied to the unbroken skin with friction. Lotions are aqueous or alcohols or polyols based solution intended to be applied to the unbroken skin without friction. And the last category is solution and tinctures that are to be employed or applied topically. These employ aqueous and alcoholic vehicle respectively. Example includes compound benzoin tincture. The next category under discussion is oromucosal preparations. Oromucosal preparations are liquid preparations containing one or more active substances intended for administration to the oral cavity or throat to obtain a local or systemic effect. Preparations intended for a local effect may be designed for application to a specific site within the oral cavity, such as gums that are known as gingival preparations or the throat that is known as oropharyngeal preparation. Generally, these preparations do include one of the following. First one is gargles. It is aqueous solutions intended for gargling to obtain a local effect. They are not to be swallowed. Second category is mouthwashes. These are also aqueous solutions intended for use in contact with mucous membrane of oral cavity. Gingival solutions. These are intended for administration to gingivae by means of a suitable applicator. And last category under this particular preparations are oromucosal drops, sprays and sublingual sprays. Apart from all categories we discussed so far, few other formulations have been kept under miscellaneous category that include otic preparation, nasal preparations, rectal preparations and vaginal preparations. Otic preparations are preparations that are to be used exclusively for the ailments or diseases associated with ear. It includes cerumen removing solutions and drops. Nasal preparations include nasal drops and inhalation solutions. Rectal preparations include enemas and vaginal preparations included douses that has been used for cleansing purposes. The most important physicochemical characteristic that has been explored under preparation and development of monophasic liquids is solubility. Solubility is a static phenomena. As we all know, whether or not a substance dissolves in a given substance and the extent to which it dissolves largely depend on the nature and intensity of forces present in the solute itself, in the solvent, and the resultant solute-solvent interaction. The equilibrium solubility of drug of interest should be determined in a solvent that is similar to the one intended for use in the final product. This can readily be done by placing an excess of drug finely powdered in a vial along with the solvent. The tightly closed vial is then agitated at constant temperature and the amount of drug in solution is determined periodically by assay of a filtered sample of the supernet. Equilibrium is not achieved until at least two successive samplings give the same result. Effect of temperature on solubility should also be monitored. Solubility studies are generally conducted at fixed temperature, preferably at temperatures somewhat higher than room temperature, that is 30 degrees centigrade, so that constant conditions can be maintained regardless of normal laboratory temperature conditions. There are many factors that do affect the solubility characteristics of active pharmaceutical ingredient. We are now going to discuss the effect of pH on solubility. A majority of drugs are either weak acids or weak bases, and their solubility markedly influenced by the pH of their environment. So, pH should be selected and adjusted wisely, and if desired, a suitable buffering agent should be incorporated in the final formulation. While we are selecting a buffering agent, there are certain criteria that are to be met for ideal or at least acceptable buffering agent. The first one is adequate buffering capacity in desired range. Second one is biological safety for intended use. Third one, no deleterious effect on stability of final product. And fourth one, it should permit acceptable flavoring and coloring of product. For many drugs, a pH adjustment does not provide an appropriate means for affecting solution. The solubility of non-electrolytes will, for all practical purposes, be unaffected by hydrogen ion concentration. In these cases, if solution is to be achieved, it must be done by the use of co-solvents, solubilization techniques, complex phenomena, or in special circumstances by chemical modification of drug to a more soluble derivative. Apart from pH, co-solvency is a wonderful phenomena that helps formulation scientists to achieve enhanced solubility of medicaments. Weak electrolytes and non-polar molecules frequently have poor aqueous solubility. their solubility can be increased by addition of a butter miscible solvent that is known as co-solvent. And this phenomena of enhancing solubility with the use of co-solvents is known as co-solvency. So, co-solvent systems work by reducing the interfacial tension between the aqueous solution and hydrophobic solute. Co-solvents generally adsorb onto the solute at the interface with water, hence diminish the interfacial tension between solute and solvent, hence results in enhanced interaction, further results in solubility enhancement. Ethanol, sorbitol, glycerin, propylene glycol, and several members of polyethylene glycol polymer series represent the limited number of co-solvents that are both useful and generally acceptable in the formulation of aqueous liquids. A number of solvents that might also be useful in the formulation of oral liquids may include glycerol, dimethyl ketal, glycerol formol, glycofurol, N-beta hydroxyethyl lactamide, ethyl lactate, ethyl carbonate, and 1,3-butylene glycol. Dimethyl acetamide has been used as a co-solvent in parenteral products, but its use in oral liquids is seriously limited owing to the difficulty of masking its objectionable odor and taste. In recent years, much more emphasis has been placed on cultivating a basic understanding of this phenomenon, with the objective of developing a mathematical approach to interpreting and predicting solubility behavior. Hildebrand and Scott have developed an equation that yields a thermodynamic measure of the coercive forces that exist within homogeneous substance. This number is often referred to as Hildebrand solubility parameter. There are several serious limitations to the practical application of the solubility parameter concept to pharmaceutical systems. The approach is restricted to what Hildebrand terms regular solutions. A regular solutions is the one in which there are no interactions between the various solvents present and between the solutes and the solvent. All molecules are randomly distributed and oriented in the system. In addition to co-solvency, dielectric constant of a solvent plays a determining role in solubility enhancement. Every solute shows a maximum solubility in any solvent system at specific dielectric constants. The solubility of a solute may vary considerably in two different solvents of same dielectric constant. Solubility profile as a function of dielectric constant appears to be similar for a solute in a wide variety of solvent system. The dielectric constants of most pharmaceutical solvents are known. Values for a number of binary and tertiary blends have also been reported, and if not, they can be calculated. Molar boiling point and dielectric constant equations may be used to estimate solubility of pure solvents and miscible solvent blends. To determine the dielectric requirement of the substance of interest, dioxane water blends having known dielectric constants are used, and the dielectric constants at which maximum solubility is attained is noted. Pharmaceutical formulations of comparable dielectric constants can then be prepared, and the most appropriate system can be selected on the basis of the solubility requirements, stability, and organoleptic characteristics. If the solubility of a component cannot be enhanced up to a desired limit, then co-solvency and other technique fails. Solubilizing agents provide a means to enhance the solubility of that particular medicament in aqueous solution. So, the first question is, what is solubilization? Solubilization is the spontaneous passage of poorly water-soluble solute molecules into aqueous solution of soap or detergent. The mechanism involves the property of surface active agents to form micelles. At higher concentration, the molecules of surfactant in water begin to form micelles, and the concentration at which circular micelles are formed is known as critical micelle concentration or CMC. At this particular point at which CMC is observed, the system depicts the maximum solubility in aqueous phase. McBain had stated, any material can be solubilized in any solvent by proper choice of solubilizing agent. This may well be true, but certain questions must be asked and answered too. These are, to what extent substance can be solubilized? How is proper solubilizing agent selected? And what are the effects of solubilizing agents on stability, efficacy, and characteristic of the final product? Whenever surfactant is to be chosen, it should be made a wise decision. It has generally been observed that lyophilic surface active agents with hydrophilic lipophilic valence values, that is HLV values, higher than 15 are the best solubilizing agents. Final selection of solubilizing agents should be based on the phase solubility studies in a manner similar to that employed by certain scientists while studying the solubilization of prednisolone with tritone. Depending on the drug's characteristics, surfactant may depict dual behavior. How? See, while non-toxic surfactants have been shown to improve the stability of vitamin A, other surfactants have been shown to have a deleterious effect on formulation components such as dyes. Another example is addition of surface active agents to drug systems has in some instances enhanced gastrointestinal absorption and pharmacological activity, and in other cases it inhibited the same. Of comparable importance is the effect that surfactants can have on formulation adjuvants. The activity of several preservatives has been found to be significantly decreased in the presence of number of surface active agents. By these examples, we can easily understand that it is not always easy to choose a surfactant of our choice, but it is to be chosen according to the need of that particular drug. Every substance has specific and reproducible equilibrium solubility in a given solvent system at a given temperature. Any deviation from this inherent solubility must be due to formation of new species in solution. So, the phenomena involved could be complexation. With complex formation, the solubility is equal to the inherent solubility of the uncomplex drug plus the concentration of drug complex in solution. Hence, complexation may result in enhanced solubility of the medicament of the choice. By use of solubility analysis techniques, the stoichiometry of this interaction as well as its equilibrium constant can be determined. This is carried out by placing excessive quantities of drugs together with solutions containing various concentrations of complexing agent in well-closed containers. Containers are agitated at a constant temperature until equilibrium is achieved. Adequate samples of the supernatant liquid are then removed and assayed for total concentration of drug. The extent to which the solubility of drug can be increased is limited by the solubility of the complex. In other cases, the limitation may be imposed by the solubility of the complexing agent. As far as pharmaceutical investigator is concerned, the major concerns are how much drug can be put into solution by a specific complexing agent and how the resultant complex affects the safety, stability, and therapeutic efficacy of the product. An example of such interaction is the complexation of non-ionic surfactants such as polysorbate 80 with the parabens that results in inactivation of the preservatives. Certain polyols have been shown to inhibit this complexation and thus maintains the paraben antimicrobial activity. Unfortunately, a very commonly used polyol, that is sorbitol, does not inhibit these complexation reactions, probably because it is too polar to partition into the surfactant micelles. A significant increase in barbiturate solubilities as a result of complexation with poly and vinyl 5-methyl 2-oxazolidone has been reported. A classic examples do include, that is, polyvinyl pyrrolidone precipitates in complex formation with a variety of organic and inorganic pharmaceuticals, and the most dramatic of which is the well-known PVP iodine complex. In addition to complexation, another type of solubility enhancement technique is hydrotrophy. Hydrotrophy is increase in aqueous solubility of many substances due to the presence of large amount of additives. There are different views on mechanism of hydrotrophy, that how does it works or how one substance can help in enhancement in solubility of a second substance. Some suggest it as a simply another type of solubilization, with the solute dissolved in oriented clusters of the hydrophobic agent. Another group feels that this is the phenomena that is more closely related to the complexation. Another reason attributed could be a change in solvent characteristic because of the large amount of additive needed to bring about the increase in solubility. The exact mechanism still remains unanswered, and this phenomena has not been much explored in pharmaceutical systems. Other considerations under hydrotrophy includes that hydrotropic solutions do not show colloidal properties. The influence on large concentrations of sodium benzoate on the solubility of caffeine is a classic example. Large amount in the range of 20 to 50% of additive is necessary to produce modest increase in solubility, and it has been seen that little use of this phenomena has been made in pharmaceutical system. Let's discuss chemical modification of drug as a tool for solubility enhancement. Many poorly soluble drugs can be chemically modified to water-soluble derivatives. This approach has been highly successful in case of corticosteroids. The solubility of betamethasone alcohol in water is 5.8 mg per 100 ml at 25 degrees centigrade. The solubility of its 21 disodium phosphate ester is greater than 10 grams per 100 ml. An increase in solubility greater than 1500 fold. In general, this approach has severe practical limitation. First thing is, new derivatives must be subjected to essentially the same testing protocol as the parent compound, including biological activity studies, acute and chronic toxicity, pharmaceutical evaluations, and clinical testing. An undertaking of this magnitude can be justified only if no other reasonable approach is available. The last factor that do affect the solubility or the approach by which we can achieve enhanced solubility is salt formation. Various poorly soluble ionizable drugs can be readily solubilized by converting them to their salt form. Since salt formation can drastically alter the physicochemical properties of a drug in the solid state, which govern the solubility. For example, an anti-malarial agent 2-piperidine beta 3,6 bis trifluoromethyl 9-phenanthine methanol is only slightly soluble in water. However, its lactate salt is approximately 200 times more soluble. This enhanced aqueous solubility is attributed in part to the decrease in crystal lattice energy as indicated by a reduction in the melting point. Furthermore, if a particular salt form cannot be isolated because of its high solubility, the desired aqueous solubility can be achieved by in situ salt formation. This is accomplished by using an appropriate acid or base to adjust the pH level while formulating the drug product solution. So, students, let us now summarize what we have learnt in this module. Liquids are versatile dosage form that can provide drug delivery to every part of the body. The soluble medicament provides better absorption, uniform distribution at the site of application. The solubility of actives is the most important parameter to be taken into consideration while we are formulating monophasic liquids. So, various parameters that influence the solubility of drug helps the formulator to select best excipient for development of safe and effective dosage form. Thank you.